Hello, everybody. Good morning. How are you all feeling today? My name is Jenny Pchelinski, and I am the Business Development Manager here at Cytiva. And today, I support our cell lines, so today I'm going to talk to you guys about engineering cell lines to help with viral vector challenges. Awesome. Sorry, first time with that clicker. All right, so here at Cytiva, our mission is to advance and accelerate therapeutics. We have a strong vision to have a world in which there's access to life-changing therapeutics for everybody. As you may know, Cytiva is part of the Danaher umbrella. You may recognize some of these companies from your past or present. They are known as our brothers, sisters, aunts, uncles, and cousins. We recognize the importance of genomic medicine in the industry. So here at Cytiva, we have a genomic medicine business unit where we focus on cell therapy, viral vectors, and nanomedicine and nucleic acids. For the purpose of this discussion, I'm going to focus primarily just on viral vectors. So jumping into the gene therapy market. There has been a significant increase in gene therapy, with AAV being our leading vehicle. Currently, AAV accounts for about 57% of active gene therapy delivery. This is significantly more than lentiviral, non-viral, and other platforms. Then from a timeline perspective, you can see that there's been a significant increase in approvals for AAV, especially within the last 2 to 4 years. While a limited number have faced some commercial challenges, many of them have had a lot of meaningful success. And just letting that sit in, because I think it's important that we focus more on the good news. And so while we have had meaningful success, we aren't without our own challenges. Some common topics that tend to come up tend to be around cost and purity. So those are the 2 topics that I'm going to dive into today. So decreasing cost per dose. So many times in the AAV market, we compare AAV to the monoclonal antibody space from a maturity perspective. But AAV is actually pretty young in a bioprocessing perspective. Here we have mapped manufacturing cost per dose in a log scale over the years, with antibodies in blue and AAV therapies in green. As you can see, the monoclonal antibody space started off with a very high manufacturing cost per dose. And over the years, we saw a decrease, so much so that we've seen about a 1,000-fold decrease since the late 80s in manufacturing monoclonal mAbs. Similarly, in AAV, we started off with a significantly high manufacturing cost per dose. Ultimately, we focused just on rare and ultra-rare diseases. Over the last 10, 15 years, we have seen a significant decrease in manufacturing cost per dose, even though it doesn't necessarily always feel like it. With this decrease in cost, moving into more prevalent diseases has become much more viable. So with a decrease in cost, we can then move to more populations that are outside of just the rare disease space. In 2020, we saw that 50% of clinical trials were focused on prevalent diseases in the gene therapy market. And just 5 years later, we saw that number increase to 63%. So while cost per dose has been a main topic, I'm going to jump into purity. So AAV impurities, I'm sure you all know them. So we have our full capsids. These have our therapeutic gene of interest. This is what everybody wants. These are the popular ones in the groups. Everyone's talking about them. We then have our empty capsids. These are our AAV without anything inside of them. They pose risks of increased risk of immunotoxicity, therapeutic expression, as well as reduced transduction. We then have our partial genomes. These pose similar risks to that of empty capsids, except these have truncated genes in them. This poses challenges not only just with purification, but also questions of what truncated genes are held within them. And I'll get into the purification part a little bit later. We then have our partial and residual DNA impurities. So these pose similar risks to the empty capsids, as well as potential risks for genotoxicity. Just as a reminder, the FDA recommends less than 10 nanograms per dose of residual cell substrates. So now that we know what the players are in this game and what their risks are, let's go to where do we get them. So we're going to go down to the production methods. So in this case, I'm going to focus more on producer cell lines and transient cell lines. So you all know about transient cell lines because this is what a majority of the market is using today. This is typically a triple transient transfection where you have 3 plasmids that have all the relevant AAV components transfected into the cell, where you then get expression of your AAV along with some of these players that we don't want, known as impurities. Transient transfection, while very flexible, poses its own challenges, such as scalability as well as batch-to-batch variability. So with transient transfection, you can also do things such as cell line engineering to improve them. We have recently launched a modified transient cell line where we have disrupted a pathway that limits the amount of encapsulated host cell DNA below the limit of detection. And while you can engineer your transient cell lines, the most productive way to limit impurities is to move into a producer cell line. So a producer cell line, rather than doing a transient transfection, you are stably integrating all the relative components for an AAV into the genome of the cell. You then select your cell based on productivity, and you choose that cell to scale up to become your producer. You no longer have the batch-to-batch variability that you see within a transient cell line, as you have a fully stable cell line. With that, you limit impurities just from the stable integration. You don't have to worry about encapsulated plasmid DNA or free plasmid DNA because you're not introducing plasmid DNA into the process. You can then also put the same modification that you would in your transient cell line as we have, where we modified our cells to limit the amount of encapsulated host cell DNA. We also have reason to believe that moving into a producer cell line would then limit, reduce, or better control partial genomes, mainly from the fact that you don't have the batch-to-batch variability. You're selecting based on how your producer cell behaves. And with that consistency, you are also able to better learn about your impurities and move into a more consistent process downstream. So on the downstream in purification, let's go through the process. So just as a note, this is not inclusive to all process-related impurities. So we talked about our AAV cell line development. So then moving to the seed train and batch production, we then get our full AAV as well as our impurities that we've been discussing of the empty, full, or empty partials and encapsulated DNAs. We then remove our free DNA through harvest and clarification. We then move through the capture and the polishing step, where in the polishing we enrich for our full capsids. While we do partially remove some impurities, this is not a full separation. As we move through the sterile filtration into the fill and finish, ideally we have just full capsids. But the reality is some impurities still lie, and some main challenges that people talk about is the partials and getting the separation of the partials from the fulls. So what do we do? I bet you can't guess what I'm going to suggest. I think cell lines are a great way to move into limiting these impurities, not just with stable, but also with transient, doing any type of engineering to your cell line. But that's just the beginning. So what can we do, and what has been done? So we've already seen improvements in titer from a transient perspective. We've seen through process optimizations. From packaging efficiency, you can move into doing any type of cell line engineering or selectivity, or if you move into a stable cell line, you can also do some optimizations with perfusion. We then have our stable production. So when you do stably integrate the genes that you need to make AAV into your cell, you're not introducing some impurities like plasmid DNA, so you therefore don't need to purify them out. And then quality. So you can improve quality by doing any type of gene editing. So an example would be something that I had mentioned, such as doing a modification to the cell line to limit the amount of encapsulated host cell DNA. But that's really just the start. There's many more things that we could do that just haven't been exploited. So in conclusion, we have made significant progress in the AAV field. We can see we have so many products that are used for production and purification out in the field. For trending, right? So we're all talking about understanding these, like how do we better purify and control these impurities? And just to kind of leave with just some questions to think about, it's basically, what the conversations used to be all about empty capsids, when now there's a lot of subspecies that pose significant challenges. So what do we do to eliminate them? From a process perspective, how can batch-to-batch variability affect purity? And then also for technical solutions. So similar to like cell lines, what can be used in combination with cell lines to improve robustness? But with those questions that I gave you, I will also be taking any questions if you have any. Thank you. I have you all convinced? No.
What range of purity do you currently see for AAV factors?
For like in general?
Yeah.
That is a really good question. Find me after, maybe we can talk a little bit more about it. I don't have any data on me to share, but yeah, that's a great question. I think it really depends on the process you use for purification, because everybody has their different processes. For modifications, we have seen, even when we modified our cells for host cell DNA, we haven't seen any encapsulated below limit of detection, I guess is what I'm legally supposed to say, but we've eliminated those impurities. So it would vary on what's been done and what you had in mind or moving forward. And I'm sorry, I hope that answered your question. Nope, we didn't see any reduction in yield. So the modification we made actually had to do with the apoptotic pathway, so we did do a lot of thorough research to make sure that no yield or anything was affected. Awesome. If there's any more questions, please, I welcome you to find me after. Thank you.